Our next speaker is Nancy Beluk. Nancy has been working in the field of MRI for 34 years. Starting out as clinical staff, she eventually moved on to GE Healthcare in advanced application as a marketing product specialist. Today, Nancy is focused on MRI research, working at the University of Pittsburgh and the UPMC Children's Hospital of Pittsburgh as a research coordinator in the Pediatric Imaging Research Center. She manages several studies which hope to derive a better understanding between brain dysplasia in children and the potentially adverse neurodevelopment outcomes that may occur as a result.
Hi, everyone. Oh, there we go. Oh, no party hats. So I have to say, I am not a manganese girl. I am not a lead girl. I am an imaging girl. But when an opportunity arises to work with someone such as Dr. Haynes, you jump on it and you say, count me in. So through name in the hat, sadly she picked the short straw, got me. And I sometimes believe my job is to disrupt her conference calls, which I'm really good at. Am I supposed to be hitting like just left, right, down, up? She was practicing. There we go. Okay, so as Erin showed you, we came in as the University of Pittsburgh in the CARES 2, the second part of this. We are the, it was on and went off. We are the neuroimaging part of it. We are actually looking at the brain. I'm going to break this all down, hopefully in a sequential type of understanding matter. We're going to go over anatomy, metabolism, organization, function. When I say organization, I'm not talking about how do they organize their home or their desk. I'm talking about how the brain organizes their thoughts and processes. We're going to talk about the function of the brain, how it's doing and using and how it should be processing data, but with the manganese. With the manganese, how is that affecting it? And then we're going to talk about connectivity, and I'll explain that in a little bit too. So we talk about the MRI protocol. So what we're going to do is we're going to look at the brain and we're going to go through all of these tonight. Quickly, I promise you. So when we talk about neuroimaging, we're talking about the structural anatomy of the brain. One of the big things we talk about is T1. Now, obviously, I don't expect you all to understand what I mean by T1 imaging, so I showed some examples. So when we're looking at the brain, we can look at the structure. This is a brain. It's a sagittal. The person is looking this way. This is actually the eyeball, sinus. This is the brain itself. Nice sulci. Gray matter is gray. Nice of it to do that. White matter is less gray. This is a coronal image, so the person is looking straight at you. So now we see their two eyeballs. We see their sinuses, front of the brain. And this is actually the brain cut off. This is what we call an axial. So the person is looking up. The nose is actually pointing up there. There are the two eyeballs, things like that. So this is a type of imaging we do every day. We do for patients, subjects, to look at structural anatomy. We can also look at pathological processes of the brain. This, T2, kind of easy names. So you can see now that the gray and white matter have kind of changed their look. It's now dark and reversed is the white matter is now dark, the gray matter is now less dark. Fluid will be bright white. So right now when you see the eyeball, you can see the lens in there. It is bright white. It is fluid. Same thing here, two eyeballs, two eyeballs. We also see the fluid that's around the brain. That fluid is really important to keep everything clean and flowing and refreshed, if you will. So, big thing here is manganese. We're looking at the deposition, where it deposits, kind of goes with your question. So Erin very nicely told us that it deposits in the globus pallidus. So you would think we would have to be looking at the pathological process of the brain, but it didn't turn out to be that way. We're actually looking at the T1, that structural image, and we do see these heavy deposits of manganese. Now, just to go over the brain region that we were talking about, the globus pallidus, it is part of the brain that controls motor and function and things like that. So when the people are at Marietta and going through the balance testing, again, we're looking at motor function, and then we're going to bring him in and image those areas that are responsible for motor function. So this little line down here seems kind of insignificant, but it does have some meaning to it. Manganese leaves the brain more slowly. So bad because it deposits and stays there, but good for us because it's not something that we need to image these kids within 10 minutes of being exposed or something like this. This is something that we can bring them in, and because we have hair and blood and nails and things like that, we know how much they've been exposed. We can correlate it with the structural images. We know it's still there. So manganese deposition will hit right here in the globus pallidus. So again, the same images I was showing you, we've just window leveled them, so this brightness kind of becomes more enhanced. Sagittal, you can see it there. I think the red arrows help. I don't need this. Kim put those on for me. So you can see where the manganese deposits. So these are the areas when we do structural imaging on these kids, this is where we pay extra attention to. Had to do the science stuff. So the other thing we're seeing when we do the structural imaging is volume changes. So we take these beautiful images, we throw them into another program, and now we get these actual volume images of or volume data of the brain. We can cut them up like it was a pathological, breaking down autopsy, but I won't even, if Aaron can't explain the dots, I'm not explaining the dots. So what we're looking at is areas of the brain that we know how they develop. We know how they develop at each point in a child's life. We can then put these kids' brains compared to what we call normal. No one's normal. And we're seeing, actually, see this lovely blob? Pretty obvious. That area, the volume of that area is smaller. It's diminished when they have exposures to lead and things like this. So this was all learned in the CARES 1. This has been learned in other studies. In fact, I think there's been a couple studies, 2008 to 2010. These now we know where we're going to concentrate our efforts. So metabolism, that was one of our bylines too. We do a thing called MRI spectroscopy. I could have jumped right into MR spectroscopy, but I thought maybe I'll stop and just kind of explain it for a little bit. MR spectroscopy is a way of looking at the chemical composition of brain tissue. It gives not nice images, it gives squiggly lines. Squiggly lines don't mean a whole lot to people, but in science it means huge amounts. You can do spectroscopy of metals. I know they use it in chemicals to look to chemical plants or steel plants to look at the chemical composition of a piece of steel to see how perfect or how imperfect it is. We can do that in the brain. So we get the anatomical images, we put this nice little square voxel on it, and it tells us what we find in there. So, again, they're squiggly lines, but people have figured this out. So we're looking at things like choline, creatine, glutamate, NAA. So NAA is our big one. We can see lipids and lactate. Now, why is that important? So we now just told you that we see these volume losses in the frontal lobe. So from the CARES 1 study coming to the CARES 2, we now place a voxel, this box, over the frontal area of the brain. Okay, again, person's looking this way. This is just a demonstration. So I didn't figure it out. I don't know who we is, my mouse. These guys figured out where they wanted us to look, where they wanted us to look at the chemical composition of these three regions of the brain. We know the frontal lobe is going to be important because there's volume loss. We know that the cerebellar region is going to be important because that's part of the motor area of the brain, motor cortex. I'm looking for one thing because I know there's an important thing I want to say. Another part we're looking at is the basal ganglia. Now, it's not a word we've thrown out. We've been throwing out globus pallidus, but the basal ganglia is the area where the globus pallidus is, thalamus is, and this is where we know that we're seeing these, I'm sorry, I thought you had it on it. These areas of manganese deposition. So we get this nice voxel, it's not really yellow. Place it over the brain, and we get these spectral images of that area. So what this one is, so here's that spectrum I was telling you about, giving us the chemical composition. where we're placing these voxels. Now, these were all figured out, again, in the CARES 1 portion of it. So when this came to us in neuroimaging, it was very easy because these guys had laid it all out for us. So these were areas that they were looking at, and they're looking at white matter, they're looking at the myelin sheath, so the area around the white matter. And so, yeah, so we are actually seeing declines in NAA. Organization. As I was saying, organization is not about how they organize their desk. How do they keep their schedule organized? It's how does the brain organize? How does it keep them organized? So we do what we call diffusion imaging. Again, don't expect anybody to understand this, but we're mapping the diffusion of water. For any nerds out there, Brownian motion, if anybody remembers that from science class in college, that's kind of what we're talking about here. So this is the image we get. Not very pretty, but it tells us so much. It tells us how water is traveling through the brain. We put it, is it brighter for you guys? Because it looks very dull to me. Well, there are really pretty colors in there. We can put this through a program, do some post-processing, and we get images that have green and blue and red and things like that. These tell us so much. So, my cheat sheet, because I always forget which way is going which. The human brain, the body, let's say, is 60% water molecules. If we can figure out which way those water molecules are going, we know which way paths of the brain are traveling, or the pathways of the brain. So, take this image, we do post-processing, we'll very simplify it, we tag a water molecule here, and then we chase him and see where he went. And if he went from the right to the left, we call that red. If it goes from the posterior to the anterior, we call that green. We can change these. And if they're going from superior to inferior, so in this image, going in and out of the image, we call that blue. And so we can see the way these water molecules are moving. And if we know the normal pathways and how water normally travels, and then we look at these kids with manganese, we can see if those pathways are disrupted. And that's what we're thinking we're going to find. You know, lead, what are all these things doing? So, again, got to go for the scientific-y, nerdy picture. So, we have shown, or again, they have shown areas. So, again, this is just a statistic. So, negative is blue, positive is yellow, orange, reddish. So, increase or decreases of areas where we would expect these water molecules to be moving through, okay? So we have found these regions where the myelination, the process is disrupted, and that's where we're going to keep going with this and apply it to these kids with manganese. Functional. This is actually a really cool thing that we can do now. We can see what the brain is doing when you activate it. For example, when you're watching a TV show, your visual cortex lights up. It doesn't really light up, but it's being activated. It's being requested. When you're listening to music, your auditory process is asking for more oxygenation. It's activating. Okay, when you're walking, your motor cortex is being asked to do something, and so it's activating. We have a way of visualizing that inside an MRI. Now, this is not a kid on manganese or anything like this. This is just a very dramatic picture that I threw in to explain it. In this one, person's in the MRI. We tell them to hold their breath for 14 seconds. We want to see what areas of the brain are activated, if you will. The whole brain lights up because all of a sudden you're holding your breath, your carbon dioxide is going up, and so your deoxyhemoglobin so this is this. Now, the point of this is This is now breath hold at 27 seconds, twice as long. So you can see the quote unquote activation is twice as bright. So we can image functionally what the brain is doing, what area of the brain is being called upon in that task. So for this one, for this study, they decided that we're going to look at different areas of the brain. I'm not the smart one. She's the smart one. The areas we're looking at are Broca's and Wernicke. Again, mean nothing, but they are vital in language processing. So Broca is actually, and there's a big difference, and I have two things. So Broca is language processing. Wernicke is comprehension of speech. Same, but different, if that helps. These are some of the areas that we're going to concentrate on. So how are we going to do that, or what are we going to do? So for this study, they have headphones on. Yes, I'm talking to them, checking on them. But for one portion of the study, when we're doing this bold imaging, this functional imaging, we say to them, you're going to hear in your headphones a noun. It will be cup, chair, window, things like that. And they are to think of the action verb to that noun, okay? They don't say it because as soon as they say it, their jaw moves and you get motion on the images. But we want them to think it. We know that if they think it, that we can see that change. Now, they're not actually walking. They're not actually throwing that stick. They're not throwing that ball. They're not breaking that window. Yes, most kids say, break the window. When you say, look through it, no. They're like, can I break the window? Fine, break the window. They come out and they go, no, I don't want to know what you were thinking. I had a 14-year-old boy who thought of the same thing for every noun. So, I'm getting a comment about a skip. Do I skip? Whatever.
Ignore.
Skip, snitch, remind me later, or install update now. No, don't do that. So,
Remind me later sounds really good.
skip, remind.
Yeah, remind me
Okay.
later sounds really good.
Good, then I'll do my balloon animals here. Oh, you got it. So we've had some very interesting interactions with what they choose. But we are able to see that area of the brain activate. Now, ideal world, we would have an MRI that was there over their brain while they balanced. We can't do that. So this is the next best thing, and it's kind of fun. So when functional imaging was done previously with lead exposure, they looked at the same thing. They looked at language function, they looked at those areas of the brain, and you can see just as before, blue was decreased and red was increased. So these are the areas I was just telling you about, Wernicke's and Broca's. Now, Kim Cecil, brilliant that she is, wrote me a line. I tried to remember it. I cannot. So I wrote it in red to read it aloud. So what they found in this study with lead, which Erin has already told us is closely related to manganese, is that instead of efficient, this is what she wrote, efficient pruned language function in the left hemisphere, which is typical development, what they found is compulsatory function in the right hemisphere, which was atypical. So it was the fact that the other side of Broca's is in the left. It was the fact that the right side was now highlighting and showing results, was the abnormal. This is not seen in quote unquote normal brains that weren't or don't have these high exposures to lead. So this is where we think we are going to just follow that path with manganese. One of the things we were talking about at the beginning was connectivity. Now, again, this has nothing to do with the study, but there's no way to explain connectivity without explaining what's going on in the world right now. There's a big push. It's called the Human Connectome Project. The Human Connectome Project is not just Pittsburgh. It's not just Cincinnati. It is worldwide. It's global. We need to start figuring out how the brain works, how it processes. So their kind of mission statement is what I put up there. But, it's a way to navigate. How does the brain navigate? How is this doing? So I showed you the lovely images before with showing the connections of the brain. We now know, you all know, those are directional images. It's showing how the brain is processing thoughts. So now we have to figure out, if this is how water molecules are flowing, how does your thought process go from A to B? We know it has to go along these fiber tracks now. We've already shown that. That's how the thought process goes. But then how does it get from here to here and then back over there? What if there's a tumor? What if there's a stroke? What if you don't have this area of the brain? How does the plasticity, how does the brain then redirect itself and get around it? So if we can map all these connections, we then can start seeing, because we can follow those water guys, we know which way they're going, we can see when they turn, we know when they're going up north, and we know when they turn to the east. So can we now say that thought process that we say is going from green to green, it actually has to go blue to green because there's a block, because there's something in that area that is preventing it. And how is the brain adapting to all that? So when we talk connectivity, that's what we're talking about. We do structural and functional connectivity. So all of the images we are taking, the T1s, the T2s, the bolds that show functionality, we take them all together. We know, I've already shown you, that we can get these fiber tracks, pretty images. We put them on a model of a brain. We then have a connectome matrix, a map that we're going to be looking at. And we put it all together and we denote these, we put the little nodes down, which way we think that thought process is going to go. And then we get these topographic maps of how the connections of the brain are going. So that's structural. Then I've already shown you that the kids are thinking of these actions. We can put the functional aspect of the brain, what we found, on top of that. So we can map out in precise, very small voxels, pixels, where the thoughts are going, which direction they're going, and which ones are being activated at that point in time. Relaxometry. What? I know, right? So we're looking at manganese deposition. We know that the brain is water. Now, a really cool thing about MRI is that it tells us relaxation. The only way to explain this is to backtrack. MR. M stands for magnetic. It's a large magnetic field. R stands for resonance. So resonance is a vibration. You can hear vibrations. You can see vibrations. You know, if you drop a stone in the water and you see those rings come out. So what we can do with MRI is put someone in a very large magnetic field. All of their water molecules are very good soldiers. They all line up. They line up with north. You remember as a kid having those, Willy Wonka, what were those willies with the lead things, and then we got the magnet and we gave them all a beard. We could take that magnet and line up all those lead pieces. We can do that with water molecules. So that's what the magnet does. Then in resonance, if anybody's been in an MRI, it's very loud, it's very obnoxious. That noise is an RF pulse, and when that RF pulse hits a water molecule, it's like punching them. So it punches that water molecule, they get all squirrely, they start flipping around. When they're moving, we can detect how much that proton is vibrating, is resonating. Now you know what magnetic resonance is all about. We can make an image from that, I've shown you all those, but we can also get these relaxation graphs, these models. So, let me see what the next one is. Is the next one what I wanted? Yes. So, we can measure manganese T1 relaxation. Come on, Nancy. No, you can't. How do you know that's not copper or iron? Well, they do. I don't make this up. They do. But they can measure this. So they can look at the relaxation of manganese, of iron, of copper in the brain. And that's how we can distinguish things because, well, I won't even go into it because, but we do. We do. So that's kind of basically it in a nutshell. We are processing these images. We are, finding out how the brain, we know how the brain should work. We have been doing MR for years and years and years. We know what the brain of a neonate looks like, of an adult looks like, of an adolescent. Now we take all that data, normative data, and process it against this group, this cohort, and we're just looking for differences and variations. So, thank you. That's actually one of our MRs for our kids. We do themes, so there's pirates and this is the astronaut one, just to kind of make it a little bit more interesting for them, things like that. So, and the people to thank, the brilliant minds behind this, which is not me. If you have smart questions, ask Aaron. If you have the dumb questions, I'm all for it. So, thank you.